video, we will be going over glycolysis. Every second inside your body, billions of tiny reactions are turning sugar into energy. But how exactly does this happen? This is all possible because of cellular respiration. Cellular respiration is the metabolic process leading to the uptake of oxygen and releasing CO2, producing ATP. But for this video specifically, we'll be focusing on the first step of cellular respiration, which is glycolysis. Glycolysis is a 10-step metabolic pathway that takes place in a cell's cytoplasm that breaks down one molecule of glucose into two pyruvate molecules. The overall reaction is shown above, starting with the reactants of 2 ADP, 1 glucose, 2 PI, and 2 NADH+. After glycolysis, you'll end up with 2 ATP, 2 pyruvate, and 2 NADH. So, let's start with the very basics. Glycolysis is the first step of glucose catabolism. Unlike the whole process of cellular respiration, glycolysis stops at pyruvate, meaning glycolysis is an incomplete oxidation of glucose. An incomplete oxidation of glucose means the cell breaks down glucose partially, so it gets less energy compared to the whole process of cellular respiration. This whole process occurs in the cytoplasm, specifically the cytosol of the cell. Within the cytosol, glycolysis will start with 1 glucose, and by the end of glycolysis, the 1 glucose will split to form 2 pyruvate. Glycolysis will also start with 2 NADH+ and get reduced into 2 NADH. 2 ATP are also produced. No carbon is released as CO2, and glycolysis has 2 phases in a total of 10 reactions. The first phase is the prep phase. The prep phase will cover the first 5 steps of glycolysis. In this phase, a glucose molecule is converted into 2 triose phosphate molecules at the expense of 2 ATP. So 1 glucose converted to 2 GAP costs 2 ATP. And on the left hand of this slide, I have a little diagram of the first 5 steps and a little breakdown of the prep phase as a whole, and we will be going over each step accordingly. The first step in prep phase, well, glycolysis as a whole, is the formation of glucose 6-phosphate. This reaction is simply a glucose turning into a glucose 6-phosphate with the enzyme hexokinase. All that is happening here is a phosphate group is being added onto the glucose. This costs 1 ATP, and this added phosphate gives the molecule a negative charge, which traps it inside the cell. It also prepares the molecule for later energy-producing steps. The second step is isomerization. So, glucose 6-phosphate is turned into fructose 6-phosphate. So how does this all work? The enzyme phosphohexose isomerase opens the glucose ring and isomerizes by moving the carbonyl. Then closes the fructose ring to make the C1 available for phosphorylation and prepare for cleavage. All that's happening here is the molecule is just rearranged. No atoms are being added or removed. It's simply preparing the molecule for the next step, where another phosphate is going to be added. So moving on to step 3, the second phosphorylation, or the commitment step of glycolysis. We're going to start with fructose 6-phosphate, and at the expense of 1 ATP, we're going to get fructose 1,6-bisphosphate. And the enzyme used in this reaction is PFK1. So a little bit like step 1, the negative charge traps intermediates after cleavage inside the cell. And finally, this step introduces a new phosphate into the molecule, which prepares the phosphoryl group transfer in the payoff phase, which will release energy. Moving right along, we have step 4, aldol cleavage. The enzyme used in this reaction is aldolase. And the reaction starts with a fructose 1,6 bisphosphate, and the products would be a DHAP and G3P. So what's really happening in this reaction is the 6 carbon molecule is split into 3 carbon molecules, which is the DHAP and the G3P. And this is the actual breaking step of glucose. This step is particularly important because it sets up glycolysis so that everything from here on out happens twice. So even though the standard free energy change is positive, the reaction still moves forward because the products are quickly used up in later steps, which pulls the reaction ahead toward cleavage. So now the last step, step 5 in prep phase, is isomerization. So from step 4, the G3P will continue directly here, and the DHAP will be converted into G3P. So now we have 2 G3P molecules, and this is important because it ensures both molecules can continue through glycolysis. It maximizes energy production, and from here on, every step happens twice per glucose. So now that the prep phase is over, we enter the payoff phase. The payoff phase is an oxidative conversion of G3P to pyruvate and the coupled formation of ATP and NADH. The reaction for the payoff phase will start with 2-GAP, and we will end with 2-pyruvate, and this phase produces 4-ATP and 2-NADH. So step 6, the first step in the payoff phase, is oxidative phosphorylation of G3P. This step is the only redox reaction in glycolysis, and it will generate 2 NADH per glucose, and the phosphorylation uses inorganic phosphate. So as mentioned, the reaction starts with G3P, and it will end up with a 1,3-BPG. So all that's happening in this step is G3P is oxidized, it's losing electrons, and those electrons are picked up by the NADH+, which will be converted into NADH, and a high-energy phosphate is added to form the 1,3-BPG. And this creates a very energy-rich molecule. And all this information is really important because this is the first step where energy is captured as NADH. It's going to set up the molecule to produce ATP in the next step. Moving on to step 7, the first substrate level phosphorylation. So the reaction is 1,3 bisphosphate glycerate is converted into 3-phosphoglycerate. And the enzyme being used in this reaction is phosphoglycerate kinase. So basically what's happening in this step is a high energy phosphate is removed from the molecule. That phosphate is then transferred to ADP, which then forms ATP. The formation of ATP is with energy provided in a coupled reaction, and this whole step generates 2 ATP per glucose. So now we're on step 8, the conversion of 3-phosphoglycerate to 2-phosphoglycerate. So this reaction starts with 3-phosphoglycerate, ends with 2-phosphoglycerate. And the enzyme being used in this reaction is phosphoglycerate mutase. So basically, if you look at the notes I took on the top of the page, we can easily see that the phospho group is moved from the 3-carbon to the 2-carbon, and this positions the molecule so it can form a high-energy compound in the next 2 steps of glycolysis. And this step is essential for making more ATP. Moving on to step 9, we have the dehydration of 2-phosphoglycerate. The enzyme being used in this reaction is enolase. The reaction is 2-phosphoglycerate and then ends with a PEP, or phosphoenol pyruvate. And this PEP is a very high energy molecule. And step 9 is a really crucial step because it's going to set up the final ATP producing step. So now that we're all prepped for this final step, step 10, the second substrate level phosphorylation, this reaction starts with a PEP, and it's going to end with pyruvate. The enzyme used here is pyruvate kinase, and the energy change is from ADP to ATP. In this step, the high energy phosphate from the PEP, which I talked about in the last step, is transferred to ADP. This forms ATP, and the remaining molecule becomes pyruvate. This step produces 2 ATP total and generates pyruvate, which enters the next stage of cellular respiration. And this marks the end of glycolysis as a whole. And now it's ready to move on to the next stage of cellular respiration. So now that we finished going over all 10 steps of glycolysis and the 2 phases, just a quick overview. In the whole process as a whole, one glucose is going to be converted into 2-pyruvate. It's going to start with 2 ATP, 2 PI, 2 NADH+. The output is going to be 4 ATP and 2 ADH. The net yield is 2 ATP and 2 NADH, as seen on the left here, like I've shown throughout the whole video, each individual step. So if you want to go back and see each reactant and enzyme and everything, just go back and look at the pictures I provided. And that is it for this video on glycolysis. So I hope you learned something about the 10 steps of glycolysis and the phases and what goes on in these steps. And if you have any questions, please ask. Thank you guys.